Pues primero, muchas gracias a Iris, Andrews y Jennifer estar acá. Realmente un honor que uno ve las trayectorias que va a presentar Claudia, pero es una posibilidad increíble poder tener investigadores de esta calidad acá. Si quieres, esta actividad además para nosotros es bien significativa porque creo que es una de las primeras actividades que realizamos en conjunto, gracias a la venida de Mr. Andrews y Iris, entre GERO, el Centro de Memoria y Neuropsiquiatría, y BrainLab. Y creo que eso es muy importante porque somos instituciones diferentes, pero creemos con capacidades diferentes. Uno más clínico y otros más investigadores, pero creemos que la única manera de poder avanzar bien en investigación es trabajar en conjunto con los desafíos y sobre todo la oportunidad que eso significa. Pero creo que para nosotros, para mí, es muy significativo porque recibir no solamente a los invitados internacionales, sino recibir brindar para una actividad organizada en conjunta acá. Y eso creo que es algo que debemos mejorar. Tenemos mucho más oportunidades si no trabajamos colaborativamente. Y ahora le voy a dejar a Claudia Durán, directora de brindar, para que presente la invitación.
Thank you very much. Thank you again, Anders and Lily, for being here. And I'm going to introduce our first speaker. We are going to have Anders after Edith and Jennifer at the end. And after that, we are going to have a coffee break. And after that, we can talk and answer a question and so on. So Edith is a neurogenetic researcher at the University of California, San Francisco. Iris focuses on identifying biological and genetic biomarkers or markers in neurodegenerative diseases, and her work supports a comprehensive understanding of Alzheimer mechanism through integration of clinical, genetic, and neuroimaging data. Thank you very much.
Thank you so much. Muchas gracias. So thank you so much for hosting us. We're really happy to be here. My name is Iris Brose Díaz. Luego les podemos hablar en español, pero la presentación para ahora va a ser en inglés. Entonces, but yeah, I'm an assistant professor at UC San Diego. And today I plan on talking a little bit about some of the tools that our group has developed that help clinicians with an early detection of Alzheimer's disease and related dementias. And so these tools, as you know, they had mentioned, is everything okay? Oh, sorry. Okay, we're good. So these tools essentially combine genetic information, clinical assessments, brain MRIs, and other fluids that you can maybe get from the blood to essentially predict who will get Alzheimer's disease and when. And so these tools have been used and are part of like the clinical routine over in Europe and specifically in Norway. And the tools that I'm going to be talking about today have been modeled using European cohorts predominantly. And so we're kind of on a mission to, help generalize these models to non-Europeans, right? So individuals from mixed backgrounds, Latinos, which is very dear to me, right? So that's kind of one of our missions. And so I want to just, talk a little bit about just like my background. And so today is a very special day for me for several reasons. So I did a postdoc at UCSF with Raul Desigan. So many of you might know him from the Desigan Atlas, right? So him and Andres, who's here, both worked together to develop the Desigan Atlas. And so that was my postdoc mentor. And unfortunately, Raul was diagnosed with ALS during my postdoc. And so during that time, he introduced me to Anders Dale, who through him, introduced me to all these computational methods that I will be talking about today. He also introduced me to Jennifer Yokoyama. And so Jennifer really, taught me the importance of studying disease mechanisms. And she really fully integrated me at the University of California, San Francisco. And so there I had a deeper understanding of like the clinical characterization of some of these neurodegenerative diseases. But it was really working with all three, right, that I had like a growing motivation of, Raul was affected by a neurodegenerative disease, and learning the computational methods and the clinical characterization to develop methods that have true, real translational value, right? And so that's a little bit about my academic background. And so, there we go. And then I swear I'll get into the science, but I do want to get into a little bit about my cultural background, because I think it's important how our culture really does shape our points of view and how, not only how we do science, but our approaches to science. And so, so my mom is from Mexico, from Guadalajara, Jalisco. And if you think that's my dad, it's not. That's Vicente Fernandez. And then my dad is Panamanian. So my dad, he met her at the University of Guadalajara. And then eventually they immigrated to the United States. My mom was like 8 months pregnant with me. So I just like held tight, and waited for the opportune moment to be born in the US. And so I was born in Los Angeles. But I grew up in a Spanish speaking home. And I had very, very strong Mexican, Panamanian, and American cultural influences, right? A fun fact is my dad's Panamanian, but he was actually born in Santiago, Chile. Yeah, and my grandma got educated in the Universidad de Chile. So this is like super special for me to be here and be with you guys. But in addition to that, why I'm telling you this is because I have a personal investment in making sure that the models that we do develop do generalize to other populations, right? And so how I'm doing that now really is, so I'm one of the investigators of Solinka, which is the largest US-based study of Latinos in the US. And it's an aging cohort. We have imaging, genetics, clinical data. And then also, I work very closely with the University of Guadalajara, where we have this capacity building program where I essentially lead that, where we train all the the students, and we work with the faculty there. They have data collections. But realistically, part of the building capacity component is that to create these federated systems, right? These systems in which the data exists and stays either for regulatory reasons or personal reasons, right? They stay in the countries in which they are collected. We are able to then bring our tools, instead of taking the data into go to the US, we bring our tools to them. And so by creating these federated systems, it allows us to work collaboratively. So my goals here today are really to talk about some of these tools that we are currently using that would have some, clinical benefit. They're clinical tools that would allow us to essentially predict who's at risk of AD, but also specifically, we want to learn a little bit about the clinical workflow in Chile. We want to better understand the infrastructure, the health registries, and biobanks, because the reality is, like, even though we have these large cohorts with Latinos, these models do just require a lot of data. And so we really have to work internationally to be able to improve some of the work that we're doing. And then realistically, like the different countries and different cultures, there's a lot of the lifestyle factors might be a little bit different. The way that the clinical characterization might be a little bit different. So it's important to improve our models in a way that actually apply to the places that we're at. And then lastly, we want to talk a little bit about, created the federated systems here as well. And so a little bit about the actual work that we're doing. So I'm not sure, how familiar you guys are with polygenic risk scores, but essentially polygenic risk scores are one of the traditional approaches to predicting disease risk, right? And so we have these large GWASs where you identify certain genetic variances, and each individual variant, because the effect size is so small that it's not really clinically informative. And so what they have done is essentially you sum up the effects of all these individual variants to create this polygenic risk score, right? And so when you do this, then you have this distribution. Okay. So you have a distribution here on the right, where people who may be at the highest risk of a PRS, right, the polygenic risk score, would be at the right end of the tail, and people who have the lowest risk of a polygenic risk score would be at the left of the tail, right? And so it's a way to stratifying people into high risk and low risk. Now, when it comes to Alzheimer's disease, we know that AD is age dependent, right? So your risk increases as you get older. And so the polygenic risk scores don't really take that into account. And so one of the ways of actually incorporating this essentially risk factor is by modeling, rather than doing a polygenic risk model, you could do something like the polygenic hazard score, where you actually explicitly model the time of an AD onset. And you do this using survival analysis. And so here on the right, I'm just showing that, the prevalence of Alzheimer's disease does change according to your age, but also sex. And so in future slides, I'll show you how we're also working on creating these like sex dependent models. Okay, so these are, so this is essentially like your Kaplan-Meier curves from your cost proportion hazard model. And so the important here is like on the x-axis, you have age. And then on the y-axis, you have the probability of having AD or not. And so these models are based off, the idea is that even if you live to be 150 years old, right, at one point you will get Alzheimer's disease. But the reality is you'll probably die of something else. I mean, if you make it to 150, like, you'll tell you, well, we won't be there for it. But like, so what we'll do is, so then when you look at this, when you stratify people into different risk categories, if you look at the highest risk category and the lowest risk category, there's about a 35-year difference, right? And so this is important for identifying like who's going to get AD and when, right? We've also made more progress in integrating not just genetic information from this polygenic hazard score, but also including age, and then multimodal data, so genetics, imaging, and cognitive data. Here, if the odds ratio is really just comparing the green line and the yellow line, as you can see, they're like very segregated. So why would this be useful? Is essentially if you focus on, who is at the highest risk and is going to get AD in like 5 years, you may want to include people who are in the red, blue, and green percentiles, right? Because those are the people at the highest risk of developing the disease. More recently, we're focusing on standard of care tools. So in terms of the actual modalities that we're using to model. And so far, we're using ADNI right now. I don't know if you guys are familiar with the ADNI cohort, but we're using ADNI and we're integrating. So we're thinking about, okay, what's part of standard of care, right? So sometimes you can collect an image. There's like clinical assessments that you could do. And we're trying to focus on those that are like shorter, because the reality is like in some places like Norway, because AD is now so common, that it's actually really geriatric care that will be doing some of these assessments, like the earlier assessments, and not the specialized clinics, right? And so we're trying to keep that in mind, like how can we develop something that could probably be used in geriatric care or primary care that can then inform who should get, more sophisticated testing or more sophisticated biomarkers. And so we started off with just imaging, and I actually don't have just the imaging here, but we started off with imaging, which performed pretty well. Like the odds ratio was around 12. And then we included the MMSC. So in that case, that did bump the odds ratio a little bit more, right? So if you use both of these, and it performs a little bit better in females than it does in males, right? Now, if you add in APOE information, as you can see, the odds ratio is increased in females significantly, and just a little bit in males, right? So this is kind of so this is kind of what you would expect in terms of like adding APOE in the model. But we've also, because ADNI is a really rich cohort, right? And so at this point, we're using all the data and we have information from when individuals were healthy, right? So we know when the individuals were healthy, we know when they go on to have MCI, and we know when they go on to have AD. So we're actually able to model the information along that entire trajectory and use that to make our predictions. And the other thing that's really important in terms of like translating this for clinical use is that we're developing models with different combinations of biomarkers. So a lot of times the clinics don't necessarily have data on everybody for every single biomarker, right? Some people may just have imaging. Some people may just have a clinical assessment. Some people may have all three. So we are able to create these different models, know which ones are better performing than others, and then you can apply it to whatever individual that you may be assessing, right? Other things are, yeah, so we will be including like PET data, CSF data. But again, I think it's important to be able to use information that you can access from standard of care to then determine who should get something more costly like PET or something more invasive like CSF, right? Okay. Here, the only thing the only part I want to mention here is that some of the work that we're trying to do is how can we use existing data, right, to improve the prediction power in individuals of different ancestry. So this is actually part of a polygenic score. So this is kind of like going back to the genetics component. But the point is that if you model ancestry information, even by you incorporating like PCs or something like that, you can actually really improve the prediction power by like 10%, which is pretty good, right? Obviously, I don't have it here, but this is actually not at the level of the Europeans, right? So if you create a polygenic risk score using European data, apply it to a mixed cohort of Latinos, the performance is pretty poor. But if you model the ancestry information, then you can actually boost that power by like 20% or 10%. And that's pretty important. Obviously, the goal would be to be able to work together so that we can collect data and actually, include it in the models, right? But we don't want to just sit and wait for that to happen as also. So we're trying to work together, right? We're trying to create the models and the new tools to be able to model ancestry, but also work together to actually grow, learn about the different health registries and the biobanks to improve our models. Another way of doing it would be by using local ancestry. So, and then this is not necessarily unrelated, but this is a different. We were talking to like clinicians at UCSD, and we're talking a little bit about what would be useful for them. And one of the things that they came up with is like, this is kind of analogous to like the growth curves, like the child growth curves, right? And so what you have here is essentially for different biomarkers. And again, you could do this for CSF, and we could talk about that later, but here I just have the imaging hazard score and then the MMSE. But what you could see is that 10 years on the x-axis, you have time to dementia. So 10 years prior to somebody getting dementia, at least the imaging score, people don't really, the y-axis is, you can think of them as like standard deviations. And so in the y-axis, it's pretty much close to 0, right? But if it's 5 years till dementia, you could see that the standard deviation is around 2. And so depending on, we have certain biomarkers, and just newsflash, the CSF one, you can actually, there's actually differences even 10 years earlier, right? And so this is really useful because, clinicians can kind of look at these different biomarkers individually, and these are compared to like age norms, right? So you can compare this to age and sex norms. And it tells you a little bit about like the prediction of these different biomarkers and when, right? And then lastly, so our goal is to, and Anders will talk a little bit more about this, but our goal is to integrate some of these models and the prediction tools into a dashboard, essentially you can think of, and that clinicians can then use, both in geriatric care, but also in specialized clinics. And you could imagine that specialized clinics would like something a little bit more different than someone in a geriatric care. And then lastly, I alluded to this, but what is one way and the best way for us to work together in sharing data and securely would be to create these federated systems, right? And so these are actually, these are different studies in Norway, and they've already created these, and this is the nice part, they've created these systems that are already working, because in places like Norway, you can't share data, right? You can't share genetic data. So and even the UK Biobank is going in that direction, right? So you actually have to bring the tools to the cohorts. And so, so this is one thing that we want to work together on. And I know that Anders will touch base on that as well. So that's all I have for you guys. Thank you so much. And if you have any questions, I'm happy to talk at the end. So thank you.
Now we can meet the micro for the next floor.
I emailed the my slides.
Yes, yes. Computer. So yeah, forget the computer, just like.
It's on this, on the.
Yes, yes.
Perfect, thank you.
Thank you.
Ahora viene Anders.
Now is the turn of our next speaker, Anders Day. Thank you very much for being here. He's a professor of neuroscience at the University of California, San Diego, and creator of the free software. Anders Day is a pioneer in developing advanced neuroimaging tools. His work merged technological innovation and clinical application to enhance early dementia diagnosis.
Wonderful. Thank you.
Let me check.
Yes, it's
Did that work?
Oh, yeah.
Excellent.
You have the
No?
No, yes, not working. Not working? No.
Okay, that's a pointer.
oh, you can
I don't know why.
The pointer doesn't work?
No,
Okay.
what?
Oh. The red button.
now.
I see. Oh,
Oh,
but you have to.
wow.
not to put, I'm sorry, not to put that, okay, actually the.
Now it's working.
It's okay.
Perfect, thank you. Okay. Yeah, so just a recent development. So I was a professor at UCSD until about a week ago, and so now I retired there, and I am president of the J. Craig Vector Institute. So it's been a very good position to do, I think, advanced genomics and integrating it with imaging, and so that's really some of the things I'll talk about today. So just a full disclosure, so I'm a founder of a few companies in the biotech space, and I'm venture on venture patents or licensed to many of the companies in the space, and also we have research agreements and collaborations with a number of businesses, and so yep. So this my movie isn't working today because I have to send it by email, but anyway, this is really just kind of here just to illustrate. So I've been working on imaging for a while, you know, so kind of automated analysis of imaging data from the brain, and there was kind of AI before anybody talked about AI. So this is about 30 years ago we developed this kind of tools for fully automated segmentation of the human brain, and also the cortical surface reconstruction, and then being able to now kind of handle it really as a two-dimensional surface, and then registration across subjects. But this is really kind of pretty antique now, but it's still pretty widely used in the field. So more recently we've also been working on how can we essentially use advanced MRI methods to, and there's a way to get rid of this thing on top of everything. But yeah, so to kind of probe tissue microstructure and see within the voxel what is the voxel made up of, you know, so we can distinguish small spherical kind of compartments of water versus elongated cylindrical versus, you know, an extracellular, intracellular, and free water. And so this is using diffusion MRI. And it's so we've kind of developed some tools for basically both acquiring the data and also then processing the data that allows us to really do a much better job of detecting things like brain cancer, like tumors, and it's kind of relevant for the whole body, but also to really kind of get better, kind of characterization of things like, edema and other kind of, abnormal potentially lesion detection and quantification. So this here shows basically, so we can study so the tissue microstructure, really kind of what we call cellularity measure, which is really just how much spherical, kind of long T2 kind of kind of intracellular restricted water do you have, and it really just lights up cancer without any kind of contrast agents. And this is, of course, getting the cylindrical restricted diffusion is the way we kind of get at how much basically it allows us to look at kind of the properties of the kind of white matter tracts and the picture in general. And then we can all bring it together on top of the kind of the segmentations. So, oops. There we go, and we also, this is probably not that interesting to the commercial audience, but here we have extended this to be able to kind of really look for cancer kind of in the, we do whole body scans, and so it really kind of amplifies, it allows us to detect kind of the neoplasms throughout the body. And then we also have methods now for automating the segmentation of all of the body organs. But that's probably of more interest to other people than the neurology. Okay, so finally, so this is kind of the workflow we have. So we work very closely with the MRI scanner vendors, GE, Philips, on kind of how do we basically optimize the acquisition protocols on the scanner, how to make it kind of button so that, so really our goal is to democratize this kind of technology, making it not just something that's available in kind of the rich people in a few places, but really kind of make it available much more broadly to the world. And I think that's, so working with the kind of vendors to make sure that it's kind of push button, but then kind of pushing data kind of from the scanners, and then we to a device that actually can now do, and then this is now typically done in the cloud. So we have then these kind of FDA and CE cleared kind of disease specific processing apps that are deployed kind of in the PACS or in the cloud. And so we have ones for kind of for basically automatic segmentation of both the brain and kind of detection of lesions. Also have for basically identifying the major tracts of the brain. And then we also create these kind of what we call this as a NeuroQuant. It's basically the product that kind of was the FDA cleared version, if you will, of CRISPR that now we can deploy in the clinic. It gives you a report. I'm going to show you a little more about that. Now we also have kind of for whole body. But the point is that then kind of we have this AI processing kind of pipelines, kind of, but then apps that now provide kind of these AI assisted reporting workflows. So like radiologists can now take the outputs of these things and really kind of create the report. And we have automated, but then also we have ways to kind of for the radiologists to customize and then we use that for you can go to the PACS and RIS and EHR. We also use it for biopsy and surgical planning and for radiation therapy local planning. And so this is the here's an example. This is actually the data from an anatomy subject. It's like 7 different time points. And see, this was one who was MCI at baseline, and they're already kind of the hippocampal volume is kind of well below what you expect for age, but you see they're also dropping off a cliff, right? So you can actually the longitudinal data is also quite valuable. This is the kind of report that's automatically generated by this. And then as Iris mentioned, kind of now, okay, that's really more for kind of the radiologist to be able to create a report, but then goes back to the to the primary care physician, the neurologist, or whatever, neurosurgeons. But then really kind of this is more like how do you now integrate all the kind of information, and that's really more for the primary care physicians, the specialists. How do they kind of now not just use the imaging data, but also the sequencing genotype data, and then cognitive behavioral measures. Where are you now? Where you should be? What's happening over time? So really kind of having a way to incorporate this, the EHR data, wearables and sensors, proteomics, metabolomics, and so forth. Kind of like having a portal, having kind of like a platform where you can bring all of this information together for what's known as clinical decision support. right? And again, this is, radiologists don't want to deal with this, they just want to get, do their read and move on. So, and at the end, you can kind of basically have a interactive portal where you can kind of do your work, but you also can have a nice printed report that you can then, show to your patients, So this, this is just showing kind of the kinds of the data that Iris was talking about earlier. So this one, so this is bringing together these particular sources of information. There's also, of course, amyloid, tau, all kinds of other information that is also quite valuable and amazingly strong. Ultimately, if you put it all together, there's very little ambiguity left. But it also allows, but it's kind of in the clinical practice, you don't really necessarily have a very, you don't have a design. It's not like a kind of like an ADNI study where everybody is to kind of assess a certain way at a certain time. In clinical kind of practice, it's more kind of N-1, you have the information for that patient, but they, it's not necessarily kind of strict kind of a protocol. And so really what we want is to have kind of a tool that allows us to bring together whatever information we have in a systematic way. Okay, so then the other thing we're working on, which is kind of an extension of this approach, is what we call an avatar approach. I don't think I have no way to get rid of this today. No, that's right. But yeah, so the kind of we have genes to outcome, kind of predicting whether so that's a polygenic risk score, polygenic hazard score, and then there's first the biomarkers to disease or outcome. That's we talked about those, but as important is actually the way the biomarkers themselves are very strongly dependent on genes, genetics, and so keeping track of this, essentially the kind of the heritability of biomarker measures promoted by imaging, for example, only very strongly determined by genes. And so a lot of the variability in the biomarker has nothing to do with disease. It's really just how you're born. And and so if you happen to have the parents who really like large hippocampus, you're going to have a lot more kind of atrophy before you look at normal, right? So I think it's really, so the point here is really if we can build in to our the decision support tool the knowledge of what's normal for you, based on your genes, not your age and not just your age and sex, but really your entire genome. And so if you can explain 80% of the variants, then you can make the biomarker much more valuable. So that's kind of our next step, and we're making some good progress on that. And then finally, I wanted to mention, again, this is kind of an extended version of what Iris was showing. So we have kind of a number of research studies and kind of biobanks that we're working with. So I'm PI a couple of studies in the US, ABCD and HBCD, just following kind of kids from birth and then the teens. First we had ADNI, and we started ADNI many decades ago, and then the COVID study, and then the Solinka study that Larry talked about. And we have another, so we have a number of different studies in the US. We also have a lot, I'm a European of origin, so we have a lot of data from Europe. And biobanks, so we have about 30 million subjects, about 2 million with genetics. But so that's an incredibly powerful database for us because we have lifetime we know the health outcomes and all kinds of the school records and everything is kind of tracked, right? So it's very different from the US. But the downside is that it's not very diverse. Diverse in Europe, but that's still very small in the world. So so that I think this is one of the motivations for us. Like, hey, can we now broaden, make the sample much more diverse, and both in terms of genetics, but also in terms of environment and social factors. And then we also kind of a smaller number now of studies where we really have kind of health data, kind of both in US and Europe, but really for where that would be more kind of place where we can actually apply these kind of decision support kind of tools. Here we can learn, we can build a database. Here we can actually, so we have a number of studies that are funded in Europe that are actually focusing specifically on the application of these methods to actually in clinical routine. And finally, I wanted to just this is, I think, why we're here. And so looking at potential opportunities for collaborations in Latin America. So federated analysis across research studies and national health registries and biobanks using secure sovereign cloud solutions, and that's what we were Iris alluded to. So we have now developed technologies for this, and that's we need to do that in Europe because most of the European countries do not allow data to leave. And so the processing, you have to bring the analysis tools to the data rather than the other way around. So this is now, so we can do, we can bring the data, the processing pipelines to the data, and then we can basically pool data across studies as aggregated data. And then the other thing is as I said, is secure deployment of the tool, AI tools and decision support solutions in clinical routine. So how for use by both specialists in hospital, but also for use by primary care physicians. So in Norway, for instance, now because people are there's a lot of older people, and so many the incidence of dementia is quite high, and so if anybody is more than 65 years old, the rule now is that they have to be they're managed by the primary care physician. And same thing, prostate cancer, very common diseases with age. It's basically the first line of is the primary care. And I'm sure that in most socialized medicine settings, it has to be because it just it becomes as the population ages, the only way you can get the all that. So they need tools now that actually are because most of the primary care physicians don't have the time and the expertise, and so I think bringing the precision tools to them and not making them much harder is important. Thanks. So that's those are my slides. Thank you.
Now I'm going to introduce Dr. Jennifer de Goyama, my colleague and friend and the leader of the genetics and everything in our Red Dot cohort. So here, Jennifer, Dr. Jennifer de Goyama is from University of California, San Francisco, UCSF. She's leading everything related to the genetic things in our Red Dot cohort, but also she's leading researcher of brain aging genetic at UCSF. Jennifer explored how genetic variants influence vulnerability to neurodegenerative diseases. Her integrative approach bridged molecular data with clinical insight to advance precision medicine. Thank you very much. Thank you very much. Okay.
Let me check the remote control.
Okay. Thank you very much. Okay, so you really do have to put it low, huh? I'm gonna put this. Okay, perfect. So I guess I'm the genetics arm of the of Udis's scientific family, so the genetics module. I'm going to tell you at a very high level about some of our work and then how we're thinking about utilizing genomic information to both enhance our understanding of biological etiologies of disease as well as enhancing clinical care. So these are my disclosures, mostly related to funding. I do also want to highlight that I'm now the founding editor-in-chief of a new journal in the Nature portfolio, MPJ Dementia. We have a number of
very relevant collections, now recruiting papers. And so if anyone in the audience is interested, we have very specific collections, including one on imaging and genetics being led by Dr. Luke Bonham, also from our family of imaging geneticists, as well as one focused on diversity in dementia research. So if anyone is interested, please let me know or contact Andrea or Claudia, they can connect you to me. We're happy to invite you for the submissions. We can waive the publication fee and it will immediately become open access. So these are all peer reviewed. The only caveat is we're brand new, so we don't have an impact factor yet, but my plan is that it will be high. So at a high level, and definitely along the themes of what we've been hearing about today, our lab is really interested in thinking about how variation across the human genome impacts the brain and how it develops, ages, and potentially has selective vulnerability to different types of neurodegenerative diseases resulting in dementia. And so we can use a lot of the same tools that you've been hearing about already, studying genetic variation, looking at imaging of the brain, as well as looking at behavioral measures of cognitive or behavioral function, and often in combination using those to try to understand why different people are particularly at risk for different diseases. And something that's very important to me is, again, the theme of today, which is really that we have to do this across many different people. We can't just estimate disease risk in Europeans, no offense, Anders. We want to do it for everyone. And so our lab is a hybrid lab. We have about 25% of our lab is wet lab, and then the rest is computational. And our tool sets are primarily genomics, transcriptomics, looking at gene expression, primarily in the blood, as well as some gene expression in brain, and then neuroimaging. And I'm just showing you some examples of other papers that we have focused on Alzheimer's disease and frontotemporal dementia. And so we could use these types of studies for both biological understanding as well as biomarker discovery. And the goal is for these types of discoveries to then help give additional information that can be integrated into the translational tools that you heard about in the last two talks. So among my many roles, I have a lot of leadership roles at our Memory and Aging Center at UCSF. And these are anchored in kind of thinking about how we can leverage biospecimen data as well as genetic information to enhance both research and clinical care. These are some of our Alzheimer's Disease Research Center. We have a program project grant focused on frontotemporal dementia. REDLAT is the study that I get to work with many of those in the room with, where we're studying AD and FTD. And I also am part of a Tau Center Without Walls, which is really focused on the biology of Tau metabolism. And so in all of these projects, I'm trying, it's a lot of hats to wear. And so we're always trying to think about how can we have a common theme that anchors all of this work and then addresses each of the specific goals of different people. Because, maybe not surprisingly, I have worked with a lot of different disciplines. So people that are clinicians, people that are data scientists, people that are basic biologists, only working at the bench in mouse models or cell models. And our goal is really to enhance the ability of any of these people to interact with genomic data in a really well-informed way. And for me, that means not just putting an analysis, data into an analysis pipeline, but actually understanding what the biology of the disease is and what the clinical manifestation of the disease is. And by translating across these different domains, we think we can learn more about each aspect of disease that will ultimately help us identify better biomarkers, better cures, and better treatments for patients. So these are different examples. Like, as you can see, this is made by Ale Castrita, one of my longtime research assistants and data scientists. And the idea is that each person is going to use the same kind of data in slightly different ways. And so we've been really trying to develop different mechanisms to create automated reports. They're not quite as pretty as the ones that Anders and Iris were telling you about, but we're slowly getting there. And the goal is, again, to think about how we can create open, scalable resources that can be applied to broad data sets, and then used to develop very user-friendly reports that can be used very specifically for different use cases. So this is just an example of our latest pipeline for, this is like, why is any neurologist going to want to know every step of this genetic data processing pipeline, right? They don't need to. As long as they know that the data was collected in a high fidelity way, it was processed in a high fidelity way, and at the end of the day, we can give information out that gives them the context of the genomic data and also the caveats or limitations of that data. These are some of the team members that are working on this, and this is the example. We're trying to create a GitHub where we can have all of our pipelines automated or like clearly annotated so that people can come in and use them and then adapt them for their own uses as well. So I'm going to zoom in just on the bottom part of Ali's pipeline, which is really highlights 3 different types of reports. And we think of these as 3 of our main use cases. So sometimes people want to know about the annotation of variants. So we have like a lot of short read whole genome sequencing data in the Memory and Aging Center participants. And people are like, I want to learn all the variants that are in MAPT, the gene that encodes tau, because I want to learn about the biology of the tau protein. And so we can create a report in orange for people that are basic scientists that are then going to go and look at these really weird rare variants in MAPT and model them in their biological system to understand how, for example, a particular variant in a person with a primary tauopathy resulting in progressive supranuclear palsy also impacts the cleavage of the tau protein. On the other hand, a bioinformatician, maybe like Iris, for example, is like, I just want all the variants for all of the data and I want a diagnosis attached to it. And then I'm going to put it into my PHS system and I'm going to help create a new polygenic score. So we might make a report that's the green version. And finally, we have our clinicians, who I know are many in the room. And these are the people that, are thinking about a specific patient or set of patients who may, for example, share clinical syndromes or neuropathological features, and they're trying to understand what is the same versus what is different in any of these given patients, and can we learn about the genetic basis of their disease. And so my lab is really particularly interested in rare variants. So these are variants that occur in a very low frequency in the population. And we think these are exciting to study because they can give us new insights into biology. And so I'm just going to highlight the kind of examples of papers or collaborations that have come from each of the types of data that I just described. So for example, we worked with Amy Gao's lab to identify rare variants all occurring in a very specific little motif. It's like a, it's a K-FER motif, so whatever, how many letters is that? 5 letters. 5 amino acids worth of base sequence. We found there was an overabundance of really potent mutations that changed the biology of the way that a particular protein, TSC1, is basically metabolized by cells and seems to increase risk for primary tauopathy. And so in this way, we were able to contribute to their work that's really characterizing this particular biological pathway that's a new way that some people may be getting disease. In a different example, we did a kind of more traditional case control study where we were looking at a lot of healthy older adults from the Alzheimer's disease sequencing project, and then comparing them to a bunch of patients that had primarily early onset Alzheimer's disease that was non-familial or frontotemporal dementia. And Nick Cochran led this study, which is a collaboration with Hudson Alpha, where we basically found enrichment of a lot of super rare variants in a gene called TET2. What was interesting was these weren't just variants that changed the protein that was encoded by the gene, but a lot of them were actually non-coding regions of this gene, which suggests that they actually have changes in the functional, expression or something about the TET2 protein or gene. And so actually this is something that Nick is following up at a functional genetics level to try to understand how this particular gene may be influencing risk, not just for AD, not just for FTD, but potentially even for ALS as well. And finally, we can think again at this like patient-centered level. We worked with a fellow from Atlantic Fellowship for Equity and Brain Health at Global Brain Health Institute, or GBHI, Adit Friedberg, who's a behavioral neurologist, super interested in characterizing genetic causes for atypical or very early onset forms of frontotemporal dementia. And one of the cases that she saw with Bruce Miller, our division chief, was an individual who had a lot of white matter changes. And so it turned out that this person, this is the image of this individual, because of this preponderance of white matter change, we were very interested in thinking about whether there may be leukodystrophy genes that are particularly relevant. And so our colleague, Marissa Ramos at UCLA, who works with Dan Geschwin, basically screened all of the whole exome sequencing for this participant, and through a very specific set of filtering that was specifically focused on leukodystrophies, she was able to converge on a particular variant in CSF1R that is very likely resulting in this particular individual's disease. And this is important because CSF1R is known in other contexts to be, at least in theory, a somewhat treatable genetic cause of disease. It's not ideal treatment, it's bone marrow transplants, but it is certainly slightly more treatable than an irreversible neurodegenerative disease. And so if, for example, we were to find that somebody early on in their disease has something like a CSF1R mutation that's very likely resulting in their disease, then it's possible that in the future we may be actually able to think about ways to intervene on their disease. And so again, the punchline for us is always, this is all great, but if you're only studying a very homogeneous population of people, how are you going to interpret the same types of mutations or results in a broader population, particularly in groups that are very underrepresented in medicine and often the most at risk for disease? And so there are both methodological and I think just moral reasons to study diverse populations. We know that leveraging the genetic diversity that occurs through different patterns of evolution can enhance our ability to map genetic loci associated with disease risk, particularly for common variants associated with disease. We can also learn, and I'll give you some examples of how a given gene may actually result in multiple types of clinical syndromes, depending on the ancestral genomic background that the mutations are occurring in. And I think particularly unique to the Southern Hemisphere, and particularly in certain parts of Latin America, there are many much larger families that are available that we can leverage to identify in a more expedited way ways that we can treat disease. And this is not something that just I have noticed. This is something highly recognized in the field, despite what our government currently may be saying in many settings, is this reality that studying diverse populations is important and it matters. And we will not stop doing this research. We may just have to slightly masquerade how we do it in certain ways when applying for federal funding. So similar to Iris, I in part came into the field with the hope that one day people that looked like me or my parents or grandparents might actually be represented in studies of dementia. And so I am very lucky to be part of this particular study, which is a multi-site study across the United States and Canada, focused on Alzheimer's disease. And this is quite remarkably, it's the first North American study of Asians for Alzheimer's disease. And it took about 10 years for our PI, Lisan Wang, to get this funded. So we're hoping we can keep it going. We can also leverage now this growing network of Atlantic Fellows for Equity and Brain Health that I mentioned through our Global Brain Health Institute. And now we have our sister BrainLat here in Latin America and Chile that will hopefully further expand this reach. But we are, a lot of my work is focused here in Latin America, but we have colleagues who are now basically trying to build similar types of initiatives in Africa as well. So REDLAT, for those who are less familiar, is our US Latin American project for studying basically the genetics, neuroimaging, behavior and cognition, and social determinants of health that are contributing to Alzheimer's disease and frontotemporal dementia. The underlying premise is that the diversity of admixed populations in Latin America are very likely resulting in genetic and social factors that may result in different dementia syndromes manifesting in slightly different ways, perhaps at different ages, and with different neuroimaging features. And that may be as a result of, differences in literacy, differences in access to healthcare, differences in formal education, and chronic stress. And so this is a now very broad project, of course, led by Agustina Bañez and Bruce Miller. Claudia now is coming in to help if we can get renewed for the subsequent iteration of this NIH-funded R01. And a lot of our scientific efforts have been led by Stephanie Pina-Escudero, who's a graduate student in my lab now and former Atlantic Fellow. So this is a perfect example, right, of the type of data that we could then have those algorithms that Anders and Iris were describing be applied to this data. Like we have it all here, right? And so we can begin to think about how can we train these algorithms to look at these different types of data with, of course, the caveat that these are still all not totally representative because they are mostly people being recruited at academic institutions in urban centers. And we're trying to change that slowly though. We know that there is a lack of understanding of the genetic etiology of disease, including in FTD, as well as in AD in the Southern Hemisphere in general. And so we're beginning to start working on this. And then this review is actually by Iris and Karen is actually published now. But, we have a long way to go. And so I think by enhancing the capacity here in Latin America to lead these types of projects, to help identify funding sources to support this type of work will be equally important to help us overcome this barrier. Because we need this information, right, to be able to ultimately apply this type of tool in a way that's going to be representative, but also valid for all populations. I'm running a little over time, but I think I'll be okay. So I'm at 19 minutes now. So as I mentioned, sometimes we find that mutations that are typically, causal for a particular type of neurodegenerative disease like frontotemporal dementia seem to only occur in certain genes. And so when we study, for example, European populations, we've come across these main genes associated with FTD. And in work led by Juliano Costa Uribe with the late Francisco Lopera in Colombia, they found that when they find patients with the mutations in these genes, that not surprisingly, they all occur in European backgrounds. Because that makes sense, right? Because they've been discovered and characterized in largely European backgrounds. However, if we look at FTD, or if they look at FTD patients more broadly and take a broader look and look at all of the different genes that have been associated more broadly with different types of neurodegenerative disease, Juliana actually found a really lot of ALS genes with pathogenic variants in them, but all of the patients had FTD clinical syndromes when these mutations were occurring on a Native American or indigenous ancestral background, which suggests that there is a lot of clinical heterogeneity that may occur, and we're missing a lot of genetic causes for disease because we're simply not investigating enough diverse people. And we saw, in addition, other genes, or she saw other genes, including CSF1R, but also genes that have been more commonly associated with Parkinson's disease with these pathogenic variants in clinical patients with FTD. So finally, I just want to end by showing this kind of crazy large family. So this is an example of one set of families identified by Diana Matayana and Juliana Costa-Uribe that harbor a mutation in TAR-DBP, which is the gene that encodes the TDP43 protein. Most mutations in TAR-DBP have typically been thought of as only causing ALS. However, there's this one particular variant, this I383V, that has been associated with FTD in other case reports. And now we have this huge families that Diana is characterizing that have not just behavioral variant FTD, not just ALS, but primary progressive aphasias, people with semantic variant, behavioral variants with motor neuron disease, as well as other neurodevelopmental disorders. And so the idea is maybe we can begin to think from a statistical perspective of how we can leverage large families like this to learn about disease modifiers. So again, our goals in Red Light, or my goals in particular, are really to think about how we can strategically collaborate to enhance capacity and ultimately expand community engagement to increase representation in genomic studies, as well as other types of neurodegenerative disease studies for different populations. It's just a representation. The colored bars represent the different ancestral backgrounds in all of the different countries that we're recruiting from. And as you can see, within every country, it's a different spectrum, right, of rainbow. And so that is super exciting to me. And then there's even this East Asian, Japanese component in Brazil. And so, you know, we think we can learn a lot, and this is just the beginning. We have our next set of genomes. Our first paper is about to be published, hopefully soon. And we have our next set of genomes coming out. We'll have probably over 3,000 genomes from Red Light to do case control studies in. And of course, that will be available for research use by the community. So with that, I just want to thank my lab. This is not an exhaustive list of my collaborators, but some of our many collaborators are funding, and especially our hosts here. These are the only pictures I can find, which means I think we need to take more pictures, Claudia, because like I couldn't find a picture of you and me together, which seems weird. But I do have pictures of when Andrea joined us when we jumped in Francisco Lopera's pool in the middle of winter at the end of a fun evening. I think this was after Argentina won the World Cup. So thank you all.
Thank you very much. Thank you, Jen. So we have time for topic and after that, for the answer and question and brainstorming and the
Yeah.
Small remain.
Now we have one table, so please have questions. Las preguntas las pueden hacer en español o en inglés, como prefieran, en caso que no haber problema de traducción.
Además iría también
Sí, traduzco si tienen, si me quieren preguntar en español.
Missing Jennifer.
¿Qué le pasó?
Jennifer llegó hoy día a las 4 de la mañana.
Oh, that's. She got in at 4 a.m. I think. Yeah.
Maybe I'll just repeat the answer that I gave.
Yeah.
Yeah, so, there was a question about if people were interested in participating in Red Black, like how to do that, so, I suggested basically contacting one of the sites that's recruiting, or whatever's closest to the area, so Andrea would be the point of contact for here. And Lizzie Barron's is the other main recruitment site. There are,
As a participant or as a researcher?
as a participant.
Okay. As a participant, you can contact, here you can contact Teresita Ramos.
Okay.
She's cuando profesionales derivan paciente, una Teresita.
Yes, sorry, I shouldn't say the question for this moment. Pregunté si, no sé, para todos, que cómo tenía que ser con los participantes, porque no lo entendí muy bien, respecto a personas que ya tienen quizás deterioro cognitivo, demencia. Entonces, ¿cómo pueden participar? Me explicó un poco, no sabía que había además que tenía que hacerse con un nexo acá. Eso era un poco.
Maybe explain because Red Lab tiene reclutamiento de pacientes con Alzheimer, demencia frontotemporal, entre 40 y 80 años, en el grado moderado a leve, que puedan estar dispuestos, tengan un informante confiable y no tengan contraindicación a la resonancia. Entonces, evidentemente, estamos buscando personas interesadas en participar y si un profesional de la salud tiene algún paciente que le parecería interesante derivar, lo que hacemos acá es que la primera entrevista es con la doctora Ramos, que es neuróloga, y evalúa si los pacientes cumplen los criterios y ahí lo empezamos. Y después, obviamente, se envía un informe al derivador. Y el email lo vemos, Ramos
@Salvador. redlatchile@gmail.com.
Lo sugiero enviarlo con los dos. Así, como todos fallamos la atención, así de Teresita y el de la coordinadora es redlat. ¿Alguna?
Hi, my name is Quentin. Thanks a lot for the presentation. My question is quite a little technical for others. My background is data scientist, data analytics, etc. I'm involved in this field. So you said that there is a way of genomic-based standardization, or your goal is to standardize data based on genomics. Can you further explain how do you aim to do this, or how can you adapt?
Oh yeah, so Iris talked about the polygenic kind of risk score, right? So that you can basically do kind of a similar thing, but instead of about kind of risk of a disease, right? You can actually look at the polygenic score for a biomarker, right? Kind of so if you have something like prostate cancer, it might be a PSA level, right? So there's a large and in the kind of the brain there would be the hippocampal volume, or it could be amyloid levels or tau levels, kind of for healthy controls for ones who don't actually develop. So what Iris was showing was kind of like there's the kind of normative trajectories of what happens to you as you age, but then there's also how do you diverge from normal trajectory when you're on your way to say diagnosis of dementia. Let's say you saw her plots where you see kind of from time zero back, right? So that's all relative to now what would be normal for somebody with your age and sex. Yes. Say, well, how far are you from that? But the idea is to we know more about you than just your age and sex. Right? If I have your genome sequenced, I can now say what is normal for you, given not just your age and sex, but also your genome. And if I also knew something about your environment, or so if we can kind of essentially shrink down kind of make the normative curves tighter, then we can also we can be much better at detecting when you start departing from normal, right? And since something like 80% of a lot of these phenotypes we're talking about are herited, are kind of explained by genes. Like 80% of the variance is explained by genes for many of these. Then if you can essentially shrink down the kind of if you can make precision biomarkers, yes, now we could they can become much more useful in predicting. And you can now detect, are you really above or below where you should be? And that's really the idea, but it's really using essentially a polygenic score, but for the biomarker. So it's not just a polygenic score for the disease, but polygenic score for the biomarker now explains the variation across individuals, not related to the disease, but just kind of essentially covariates of no interest, if you will. Right.
It could be imaging.
Exactly.
So like, I think, so if I was trying, well, because I was like, oh, this is a good idea. So the way I was thinking about it was, so you could create like the genetic estimate of what your brain looks like at baseline for your age and sex. And then say, okay, if I only had your genetics and I made an estimate of what your brain looked like, this is what it would look like. And now I'm going to see what it actually looks like in real life and compare it to the genetic ideal of your brain versus the real brain.
So think of it as a digital twin is another term, right? So it's really kind of, so it's a counterfactual twin.
Yeah.
Somebody who had all of your genes, but didn't kind of have the risk factors for disease, right? So that's kind of, so it's really kind of all of the variants in your genome that really are just explaining differences in the biomarkers, but not actually related to the disease of interest, because you don't want to throw out the baby with the bathwater. So it's important. So it's ends up being kind of, that's why I kind of think of it as kind of like a mediation analysis kind of framework statistically. So you want to understand, you want to basically remove the variance from the biomarker that is just due to kind of factors of no interest. So that's really, I don't know if that makes sense or
and I gotta say it's quite a beautiful work, because right now when we evaluate data, we standardize it between age and or educational level or sex or something like that.
And it becomes really important also in the kind of in kind of admixed kind of populations, because now, like you see, like in Norway too, like suddenly like you have a baby who has kind of like the Asian mother and whatever, so like suddenly like what's normal now, and it's also kind of just being some percent this and some percent that, as Iris was showing, it actually the local ancestry matters. It's kind of like, did you happen to have inherited the variants, say, from your African or your Asian or European ancestor that are associated with, say, being taller or shorter, or kind of having a large hippocampus or smaller hippocampus. So really, we can build in to kind of, so one thing is all the kind of the GWAS, all of the kind of the tiny little variants, but there's also just these kind of admixture kind of blocks, if you will. And so basically we're building in kind of to what is normal for you, not just that you're 50% African ancestry, but because that explains very little. What's more important is actually which particular segments of the genome did you inherit from this ancestor or that ancestor. And so I think it becomes really important here in the US. It's important, but it's also, I think, important in South America, because you have the recent ancestor admixture of many, you have Native American populations, you have African, you've got European, you have kind of a beautiful mix, but and a lot of recent admixture, and that becomes, and so that actually we find that the polygenic risk scores are not very good in these populations because of this. It's so much, it works beautifully just if you look at just within Europe, but then it really doesn't work well once you when you try to apply it in the population like this. And so I think that's why It's so some of these tools become really important, but also you need to have the training, you have to have the data. And so much of what the data that exists is really very kind of European, kind of biased. And also partly because the tools don't work very well, kind of across, with that mixture. And I think that's part of our goal is to kind of have the data that allows us to kind of really train these kind of models and then really kind of have some solution that is more generalizable.
But also you could try to add it to this equation some social determinant of health, lifestyle, and so on, that is very important here in Latin America underrepresented.
Adding lifestyle factors.
Yes, exactly.
Yes.
And because those are also things now we
Yeah,
know
it's so exciting,
it explains a lot of the, as you say, like the Lancet article on Alzheimer's, right? How, there's a kind of very high percentage of the risk that seems to be associated with kind of modifiable lifestyle factors. Interesting now also with those NPIC, we work with Novo Nordisk over in Denmark, and it's now the kind of diabetes / obesity drug, right? Turns out that now it actually has a fairly strong effect on reducing kind of the risk of Alzheimer's disease, it looks like. And it could well be that you can think of it as kind of an artificial lifestyle intervention, like it makes you basically behave in the way that you're.
I have a methodology question. To build this evidence, you need a longitudinal data, or you can work with cross-sectional data.
Definitely we can work with cross-sectional. But for some things, when you want to predict future events, then it's important to know. I don't need longitudinal imaging data. I don't need longitudinal anything other than I want to know what happened to the person, right? So the clinical outcomes are important because if you're going to kind of predict for the future, your training data has to have longitudinal outcomes. Did this person, how long did you follow the person without developing disease, or when did they get
someone comes
diagnosed?
to the clinic with like mild cognitive impairment and then it's sick.
And there is no way, because in a lot of countries, to have longitudinal data is very complicated. We don't have the funding, we don't have very big cohort. There is no way to work with taking population, healthy population, population with disease, and combine different, but not.
Yes.
If we don't have longitudinal data, can we do something, or we are?
Yeah, but it's not, and then it's less about future prediction, right? So, but if yes, for diagnostic, absolutely, yes. So it really kind of, and that's, I think that's where it's really important, I think, to sit down and say what kind of data does one have, because, and then, what can you do with that, kind of imaging, genetics, other measures and.
Differential diagnosis, or like classification versus prediction.
So, yeah.
So in that case, then you would, like, for example, let's say we had the Chilean data is cross-sectional. Would you be projecting or making predictions on it based on like prior data developed in European?
Yeah, but it's not even necessarily prediction, right? So it's like it really kind of like, so my understanding is that, for instance, you will have, like what we're being told, at least like in Norway, kind of like the kind of practicing physicians, that one of the things they want to know is this person like actually are they really on their way to Alzheimer's, and how much vascular? Okay, is it really kind of neurodegeneration, or is it vascular? Because the interventions might be quite different because so I think that's what and being able to use kind of combined data to not do differential diagnosis, as you're saying, and that may be important for psychiatric. I mean, there's as you know, early on, it's kind of there are ambiguities that may be important, because of also knowing should you now that they're actually the disease modifying interventions, then knowing that should I put this person on anti-amyloid to kind of the treatment, or should I be with another intervention that would make more sense. I think by the time you really diagnose somebody with Alzheimer's or dementia, I think it's too late for a lot of these drugs to really have an effect. You can't regrow the brain, you know. So I think by the time you actually have unambiguous symptoms, then probably it's too late to intervene kind of effectively. And I think that sort of having early detection and also kind of differential kind of that's my understanding. So that's what we're being told by the memory clinics and the primary care physicians back home.
And
Just, yeah.
if we're working with like the clinics, like if a patient comes in, even with subjective cognitive impairment, right? And then they come in a year later, so that would be longitudinal data we would be able to use, even if it's just the diagnostic.
Exactly.
So we can have an image, and this is like why adding is so awesome, right? Is that we knew when people were healthy, NCI and AD. So we can use the MRI at any time, as long as we know their clinical trajectory. So we don't necessarily have to have like the imaging longitudinal, but if they came in with subjective or vascular issues or something, and then you'll know when they had a deeper or not, right? So we could use that information in our minds.
Indeed, the psychiatric is very one of the. We have, for example, we have our memory clinic. You receive a lot of patients with a long life psychiatric disorder. These people now are living longer, and when we don't know a lot of time is that the symptoms are explained by a neurodegenerative disease or vascular, and we have to try to do the differential, the diagnosis. Right. We feel that we don't have the good tool, and for the system, it's very important because at the end, this patient they live in the right, oh, what they are, and the psychiatrist say no, they are from you, no, they are from you. It's difficult for the patient, but it's also difficult for the system because when you don't know really where they are, they don't receive the appropriate care.
Yeah, and like the drug like the treatment that they actually, some of them I know who have like AD, the treatment would be essentially.
Well, what is the life expectancy of men and women in Chile?
Woman is 84.
84.
Okay, so it's higher than the US.
Move here. One comment on the social determinants of health, I think it's really important to work with, the groups and the different places because, there's different factors that might affect different countries too, like access to water or, different.
How much fish or meat do you eat?
I know, probably healthier. Oh, God. Yeah.
Pues salsa.
¿Hombres? ¿Y los hombres?
70 y...
Oh, thank you.
Men, 78, and women, 80. Yeah, that's right. So it is comparable. It's comparable to Europe and the US. It sounds like.
But we have the fact of social determinant of health are very important. Indeed, there was a study published in the Lancet that in several cities in Latin America, and even in Santiago, we can see difference of 10 years of life expectancy, even if you live in a very near community. It's awful. More than 10 years, indeed. It was a very good study published, I think, 2 years ago.
I'd like to look at that.
That would have a huge impact, right, on your trajectory, if you're like thinking about progression over age, which.
But do we know what the determinants, kind of specific determinants are, or is it just, so do we know what factors, is there something that can be measured?
I don't remember the study, but they measure some social determinant.
Okay, right, so they would do that. Socioeconomic status, like income and education, yeah.
Pollution, yeah.
Oh, yeah. Yeah, and that's actually kind of also makes, I think, kind of since, so say in Scandinavia, everything is so regulated, and so there's the variation is so tiny. but I think that's kind of, like,
Everybody has access.
both genetically and, kind of environmentally, so I think we're really underestimating kind of, the importance of these factors in a way that, and that the kind of having a broader kind of sample becomes really important, I think. Yeah.
No, it's amazing. It's not very nice. For people walking in the, I was here, I live in quite rich craft, I receive patients from the British system, and I work in a private clinic, and when you see early people, they are two different.
That's
Every day, so.
Yeah, the public system and the.
Have you tested in your tool the utility of vascular imaging signs to predict or to generate or to improve the model when you predict or you show to the physician?
Yeah, we
Considering that the imaging data, you can get a lot of vascular information with matter hyperintensities, ACL, ASL, microbleeds, etc.
Yes, and so we do have, we are looking at that. So so one of our FDA cleared tools, lesion quant, actually is kind of is picking up that. But so in terms of kind of white matter lesion burden, yes, we have that. We are working on it now. I don't have the results to present to you, but yes, we're also looking at with. Kind of in UK Biobank with proteomics and metabolomics, this kind of outside of imaging. So and again, there are kind of fairly strong predictors kind of cardiovascular outcomes that then we can also incorporate in, and of course there's the blood pressure and the standard things, but there's also actual molecular markers that we now can. You can get, thousands of them with one test. It's amazing, kind of amazingly rich, it's and so we have for like 300 something thousand, you can buy a bank of metabolomics and about 60,000, I think, proteomic kind of batteries. So it's becoming standard now, and most of the studies we're working with, so there's the imaging, kind of course, just with this 3D T1 is the standard one we do for just normal brain anatomy. And then we do kind of 3D flare, T2 flare, which is kind of getting you kind of the more the edema and kind of white matter kind of.
And I think like the practical problems, I guess, I don't know if that's the right word, but is that like it's interesting how it's not always annotated, some of the vascular issues, right? Or people who go on to have dementia, it's like they just ignore if it's subtle vascular stuff. And so especially early on. And it's interesting because even in some of these very, very, sophisticated hospitals with all these data, what the radiologist actually reports is really important for us so that we can, detect that early on. Otherwise, you're left with like a very small sample size to even model some of these
It's not consistently reported.
It's not.
That's our challenge. Right. Kind of eventually, somebody might be, you have the ICD-10 code of vascular dementia, but it's really uncommon. Like almost, but a very large percentage of the patients who are diagnosed with Alzheimer's, also, if you look at the brains, there clearly is kind of white matter and hyperintensities, and but it's not reliably noted in the clinical notes in our experience, even at UCSF.
But yeah, both the vascular and the trying to the differential diagnosis in psychiatry is things that we're actually working on. So we would love to.
But it's a challenge, kind of, because the clinical routine isn't very consistent on that.
How are you thinking about the cultural differences for things like psychiatric disease?
Yeah, it could be, and exactly. So we work a lot with the kind of the psychiatric GWAS consortium, the PGC, and that has, from all over the world, right? So, and that's, the whole host of diseases, including actually dementia.
I think it's fair to say, you should learn from people who actually know those differences, right?
But I think it's tricky, because it's not even it's social, cultural differences that are quite significant.
But part of the things we want to learn are like, what are the clinical assessments that are used here for the diagnosis, right? Because if I'm modeling using like MMSE, like that's not useful here, right? So if it's important, and I'm learning in different countries, like what people are using, and some of these have been adapted. And so we could technically apply it, but.
Do you do like AVLT or any, do you do any kind of more like delayed, do you do any memory tests that are more sensitive?
There is the primary care, but here we do.
Yeah.
Neuropsychological assessment,
Yeah.
functional assessment.
Yeah.
Because those other tests are so, they're kind of sensitive so much earlier. It's like, because, you know, the one for MMSE, whatever CDR, they really are, you have to have the disease, you know, really important to be useful, right? But for some other tests, you can really, it really seems to go, you know, 10 years earlier, you can actually already have warning signs. Yeah.
And you know, one thing I was mentioning is like knowing what is done in geriatric care or primary care versus the specialized, because in primary care, they don't have the time to do any of this stuff.
Do they do computerized kind of tests at all, or is it all kind of no?
We only do a computerized questionnaire, but now it's. And what we do here, but here we will count the votes.
Yeah.
Audio, and we are doing that regarding the audio in patient, including red light, but we are also recording audio and in the patient that we see the memory clinic and they do not fulfill the inclusion criteria for red light.
So, yeah.
We see a lot of patients that we don't know if they have Alzheimer, frontotemporal dementia, that we don't.
Exactly. What's your standard workflow for somebody who comes in with a memory complaint? Kind of, do you send them all for MRI scans, or is that the kind of a?
But the receipt, we will explain it in detail in the afternoon, but.
That's right.
That's good, yeah.
We can save it, yeah.
Okay.
Person-centered care.
What's Teresita? Oh, yeah.
No sé si hay otra pregunta.
Naive question. For example, if you have some research score, and if we are thinking in Latin America, after that we have this research score, what happened?
The risk score?
To
So.
go with primary care or?
Right, that's a good question. So, well, I think it's important to know what you want to happen. And so what is happening, at least in Norway, right, is they use this in geriatric care to know who is going to get Alzheimer's disease within like 5 years, for example, right? And so depending on the tools that are used, like if it's an imaging and genetics, or if it's just imaging, then they could use it to be like, oh, maybe we shouldn't do PET on this person, right? Or maybe we can enroll these people in clinical trials, right? So especially early, right? Who are those that are going to go on to develop the disease, right? And so that's one way of using it. Other ways could be like a differential diagnosis, right? But not with the PRS. The PRS would not be the classification, but it's pretty much like, do we want to send them to a specialized memory clinic, right?
Yeah.
And now also, if they have like mild cognitive impairment, right? If our models are saying they're likely to progress to AD, that's important information, right? Versus like FTD or something like that. And so I think what we want to think of, we think of like AD and FTD as like, even an MCI state, right? As like, you already have cancer, right? It's a different state. Right, but you have Alzheimer's disease at that time, right? And so really, you don't give them the diagnosis, right, of AD because their state is MCI, but starting to think about them as like, it's like cancer, right? So, that's kind of how I think.
So if left untreated,
yeah,
are you likely to decline? And if so, then I think kind of it would indicate kind of use of disease modifying kind of interventions, but it could also be that about it's about 50-50, right? At the MCI level, those who actually will go on to kind of Alzheimer's diagnosis in the next 5 years, and those who will not. And so we can kind of with a reasonable accuracy say, hey, this half really have a very high risk, and this have
Right.
that's a very low risk. And then I think that's kind of would then, because it could be things like, oh, maybe this person is just depressed, whatever, and then you
Yes.
don't want to be, putting them at risk for some horrible, side effects, if in fact, you could, the treatment, the approach is quite different.
Yeah.
Also vascular, it might be quite different what you should do. So I think that's kind of what I think how we would kind of, again, like, I'm not going to really re-listen to the physician saying, how could this be useful to you?
Right.
And that's the sense that we're getting, that's kind of the deciding how to manage the patient and also set the expectations in a way, because, but it's also really important for those who are actually worried, well, don't, based on our test, you don't seem to be on the, you don't, the dementia isn't imminent.
Yeah. Right. And so even for them, like for planning, right, I don't know, I think the patient might be interested, hey, in 5 years, I'm gonna get like, okay, maybe. Or also like if you're including some of these lifestyle factors, right? It's important, I mean, you can't necessarily go from like low SES to high SES, but maybe some of these other modifiable, like even diabetes, like Ozempic, right? So if someone is diabetic or something and they're on the trajectory of AD, like Ozempic might be something to consider, right? Versus other medications. So I think there's a lot of like applications. Choosing your favorite would be our.
No, I mean, I have another question because I'm not a physician. Maybe it's for you, Andrea. People want to know if they are going to have Alzheimer or some dementia.
Someone.
Yeah.
I think we don't have good data to say that, but indeed, for some people want to know.
We get a lot of pushback on the genetics, because it's something you're born with, right? And half of the people don't want to know, and their families are also implicated, whatever. So it's a much more, people find that much more scary. But if it's really early detection and actually kind of making sure that you can intervene in time, then they're much more kind of acceptance in my experience.
And also in the cases where it's like less, I guess, controversial, is when patients come in with like subjective memory complaints, right? And so then in that case scenario, then you could let them know, like, okay, well, this is where, yes, more, we're not right now, but in like a little bit, so.
Indeed, if you see there was the case of a good biomarker here without evidence, and they put in the market, and people go and do the test to know if they want. Not so much, but they try to. So I don't know, but there is some interest.
Yeah.
I have a question about one thing is to predict, is to read out dementia, but as a clinician, we have a lot of difficulty, and it's a very important question, to predict the trajectory of disease. And to see some patients with Alzheimer's disease, they go there, and so they there, and what we say now, we have to expect. To try, evaluate, to do, and that is a very important thing for the family, more or less, the prognosis, when you have the disease. Do you work with that?
Yes, and so the interesting question, because we've been, when we work with pharma and with kind of CROs, they're most interested actually in kind of saying who is going to decline rapidly, because they actually the outcome, what they use to kind of to measure the effect of the drug, the primary endpoint is often kind of the rate of decline, the kind of like or kind of clinical progression, more than the kind of diagnosis kind of per se. So then yes, so we actually show, so we work with like with Eli Lilly, particularly on this. So where we essentially can use these tools, not so much for diagnostic purposes, kind of as we're talking about today, but more to do enrichment of clinical trials. So you say, like, how can you select the patients based on all of this, kind of that will actually be the ones that are declining more rapidly, And then, of course, now if left untreated, if you're gonna have a kind of rapid decline, then you're gonna have much more power for detecting the effect of a drug. Because if half of the subjects that you're treating aren't on their way to, or are not going to decline, even when left untreated, you really are reducing your statistical power, and you're subjecting patients to side effects for no good reason, right? So yeah, so that's another. So I think that's more. I'm thinking more as a clinical kind of trials, kind of a use case, but it's also important, I think, for physicians and patients as well. Yeah.
Yeah. Do you think that also, like, different biomarkers are useful for either tracking progression versus whether, for a diagnosis, right? So, oh, yeah. Go ahead.
Yeah, no, I was just gonna say, I mean, so like as an example, I didn't end up showing it today, but we have a new finding that we're trying to follow up with actually a genotype difference that predicts differences in trajectory, trajectory, but not baseline disease risk.
That's what
we're also. So, and it's an immune related gene. And so the idea is perhaps like what we'll be applying for grants to try to show that or prove to ourselves that this could be something that we could use as a prognostic indicator. And for exactly that reason, because if we could bring people into like fast, medium, slow trajectories, that would have immense implications for.
¿Hay más preguntas?
I want to make a comment. So, for us to be to actually work together, right, it's gonna require, someone like myself who, or others, right, that I've trained a lot of students, right? And so, but really identifying people on your team who would be able to work with us, right? So, if you do imaging or genetics or any kind of computational modeling, or you're in the clinics, right? And, one of these reports are useful for you, it's really about like identifying, and I'm glad there's so many people here because it's really us working with you guys here, right? So it's, if you're interested, talk about it.
Passionate person locally who can help lead and drive the project and mobilize the effort here.
Yeah, because it's real thing, we come here and we give these thoughts and we're all very excited, but actually making this work, actually making this work, finding this working.
¿Más preguntas? ¿No? La última. Nadie se va a quedar después por arrepentido de no haber hecho una pregunta. Oye, el mejor comentario que a mí me desinhibió una vez que estábamos en una charla en Bolivia, puro neurólogo, yo creo que de esa época era la más joven, Y llegó un profesor japonés y nos dijo a todos, no hay vergüenza, que no es la vergüenza, no es por la pregunta que uno hizo, que puede parecer tonta, la vergüenza final peor es por la pregunta que no se hizo.
Do you work with blood biomarkers of neurodegeneration, and how has been the performance about that comparing with other types of markers of neurodegeneration, maybe PET scan or CSF? How has been the performance?
So PET performs really well. But again, I think it didn't perform so well to where like the imaging and the clinical assessment information wasn't useful, right? So I think we could use those standard of care tools to then determine who gets PET. Because yes, if you have PET, then it improves the prediction power by a lot. And then we also looked at plasma, but it just like performed so exceedingly well that I want to go back and kind of do a double check. So, because when things perform too well, you're kind of like, what did I do wrong, right? So
we have CSF kind of
that one,
amyloid and tau, and it does well. We have PET amyloid and tau too, although less tau, more. And then we also have and then we have the serum ones, and so it's and actually in acne there's actually
And it's funny because if we look at our model, if we have like imaging, genetics, clinical data, PET, NCO, it's like you said, there's no room for interpretation at that point. But just the reality in the clinics that someone's gonna have all that is, it's not,
But I think eventually, I think the serum ones will,
yeah.
I think, become, it's kind of expensive now, but it's, eventually, I think it will be, probably
And,
will be very generally available.
yeah, and,
But the neuroimaging.
No, so,
No,
yeah,
the
very
PET.
much report, yeah,
Yeah,
but so in the US, at least, and also in Scandinavia, whatever, I think they use, like, imaging is indicated, MRI is indicated just because also to rule out, is mainly like rule out strokes and tumors and things, so you're gonna have the imaging anyway, because you couldn't replace imaging with the molecular markers, so they become kind of on top of, typically, that's the way we kind of see it there. But you want to have the cognitive test generally, some kind of behavioral testing, but also imaging to rule out. But now you can rule in, and you can also, the same data is useful for actual to measuring.
and it's important to do cost-effective tools, right? So, we're very cost.
But I think ultimately, I think serum biomarkers will be kind of come down in cost and be very available and probably be spent probably far standard.
Yeah, and some of these biomarkers made like the plasma would be better if you're trying to detect really early on, right? Like you can see differences when people like 10 years before they actually have dementia.
I think that's the one of the challenges, I would say, it's actually for amyloid in particular. It's sensitive kind of way too early, kind of in a way, so you could be living with kind of high amyloid for 20 years and no dementia. So I think we find that kind of the combination of amyloid and tau is important. If you have just as elevated amyloid with no tau, it could just be you could, so many people kind of die with lots of amyloid on board and no cognitive kind of symptoms at all. So
combining it with the MRI, so you're able to improve that. So it's like to kind of rule out those who just have high,
You're gonna show your something about biomarker data.
Oh, cool.
Okay, cool. Great.
What's
Sorry, I really wanted to avoid the technical question. I'm going to ask.
Go for it.
How do you merge this data in the multi-level analysis? I mean, you have imaging, you have all this kind of tabular data, and numerical data, etc. And I don't know if you use language models or something like that.
Really,
So
yeah.
how do you merge all this? You have a unique model, do you transform it, feature selection, or something like that?
Good question. Yeah, so we kind of started with a kind of just a more kind of classic, old-fashioned kind of approach to a lot of these things, kind of machine learning and statistical modeling. But we are now using, kind of for a lot of image processing, kind of like convolutional neural networks, in addition to kind of our atlas-based kind of statistical analysis. We're also starting to use vision transformers, kind of those things. So we work pretty closely with NVIDIA and some of these things. And so, yeah, so we kind of start. I like to kind of understand it kind of rather than just putting it into a black box. Our approach has been more to try to build on kind of science first, kind of physics, and then I kind of use kind of the more of these AI tools more as a way to kind of mop up the remaining variant. This is just kind of more because I need to understand what it does rather than having it be kind of a black box that is kind of a magic, kind of a oracle. But yeah, so we do use all of the above. But yeah, but when we put the data together, kind of once it's turned into numbers, yes, then we just basically put it through just principal component analysis, kind of standard, and feature reduction methods, and then the proportional hazard model to see how to. So we do each domain by itself, like the imaging and the biomarkers. So we do this hierarchical thing where we basically, for each domain, we kind of put it, to turn it into kind of like a predictor based on it, and then we combine them together. But that also allows us now to take the date. If some patient has they don't have PET data or they don't have MRI, whatever, then we can basically mix and match, right? We can use the data that's available, and that becomes really important when you're doing this in the clinical setting, because then no patients are the same. They will have data acquired at different types of different types. So then it becomes really important to be able to take the data that's available and then kind of get a prediction that's and a diagnosis that's appropriate for that person, rather than forcing everybody to have exactly the same, assessments, because it's just not practical.
I was gonna say something. Oh yeah, no, so another part is like, what's really cool, he's definitely the developer, so it's really fun to work with him and like look at the code and see how it works, and it's like so elegant and like simple at the same time, but it's really cool how you can model in different cohorts too. So essentially, you can eventually validate it, and the individuals will have the same, but we can leverage like UKB to essentially create the cardiovascular score, right? And then we'll combine it with imaging from like a different cohort. So that's really important. We've also modeled the ancestry components, right? So Linko is really useful because it's a cohort that has like, Dominicans, Puerto Ricans, Mexicans, Colombians, and Cubans. And so, as Jen showed in her plot, they have different proportions of ancestry, right? And so we can actually model that in Solinka and then project that in like other cohorts, right?
No study has it all,
Exactly.
and so that's why it's really important.
Right.
We're gonna do the
We
detective
figured out
work.
how to do that, like how to like just leverage these different cohorts so that we can then, apply it in smaller. So, but learning what is important to.
Whereas, for instance, genetics, it's kind of something that, you require a humongously large cohort. And so we can train based on, like millions of subjects. But then imaging, we're not going to have in millions. And the serum biomarkers, we're not going to have in millions yet, right? So it becomes really important, I think, to kind of divide and conquer a little bit, so we would essentially train models on the kind of data we have on where we have genetics, I'm happy, I'll use that. And then I have, kind of other data set where I have, proteomics, whatever, and then you want to maximize the power for each of these analyses. But then it's on things like ADNI, we have kind of the magic where we have it all together, but it's a much smaller N. But that's okay, because now we've already reduced it from, millions of dimensions down to a few.
Yeah.
And now it's possible to actually fit the model so that's kind of strategy.
Right. And then at the end, it's like you get to decide what classifier, do you want, cost proportion hazard model, like, you've already done all that data reduction, but I get to that point.
That's kind of been our approach, and
Yeah.
there's no study that has everything. So I think that's really also part of our kind of motivation here, is that working kind of with you guys and really kind of to make make it to have more diversity and kind of test generalization. But that's, I think that we can, and its European studies have real strengths, but they also big weaknesses. And the biggest one is really that we don't know how well it generalizes to the rest of the world.
And we also know that like when you're modeling and you're including people of different ancestries, you actually do improve the prediction in people with different ancestries. So even if they're like, UKB and whatever diverse cohort is there, which is mostly like African and Indian, I don't know.
Hardly any. It's really just incredibly, and also partly because
there's no
they recruit. There's also very biased recruitment there.
Right.
So it's not even kind of, it doesn't even match the UK kind of demographics at all. It's very kind of
So if I understood well, you have to create a new model with an underrepresented population, or you are going to improve the model, or how is the?
I see it mainly as expanding the kind of extending and to make sure that the training includes enough diversity to kind of really but it's not, so it's I wouldn't.
But I mean, if we have the imaging data, we could just ask the first question is how well does it perform, right? In your cohort. I think it would be important just to see what that looks like, but, you know, developing methods that can leverage data that we have to improve the prediction now, but realistically using the data to like on its own to create it.
I think that it's a very important point because the question is that when the participants that have all the data, there are a bias. They have some indeed like a brilliant participant, a different participant that the participant that you are not able to have all the data, and that is like a factor that maybe when we work only with participants with all the data, we are missing some important factor. We are working with a very select sample.
Yes. I think kind of missing this, we have to handle that, and as you say, kind of the missing this can was informative. Because it's not independent of outcomes, and so that's and so it's important to take that into account. Yeah.
Yeah, because even like something like UKB, I mean, like if they got dementia, they like stopped. They didn't actually.
There's hardly anybody
stand
with
back.
imaging. So if you look at kind of, you know, there's a kind of good number of UK Biobank individuals who actually have the diagnosis of dementia, but almost hardly any of them have imaging, which makes sense because, you know, like actually getting, you know, traveling around the UK to and then getting scanned. You know, it's actually, so you have to really go out of your way to recruit and actually, you know, assess individuals with dementia. You know, if it's, so of course we could have imaging much earlier and then it just long before symptoms, that's one thing, but hardly anybody, we have essentially nobody in with dementia diagnosis in it's only like 70 or something out of 65,000.
87. And even fewer with vascular dementia, right? So I was like, okay, there goes my training model. Like, but you know, this made me think, like, you know, we actually just came from Argentina and we're going to Brazil and Colombia. And, you know, you guys are part of that same network, right? And so really like training, I know like the ancestry proportions are a little different everywhere you go, but like some locations might have more PET, right? You guys might have more like psychiatric, vascular information, and really working together will be important for creating these models, right? So we can train them elsewhere and validate, and if you guys have fever PET or something like that. And so thinking like, you know, in terms of Latinos, right? It's like really working together to do this, yeah.
Yep.
Thank you very much once again for being here. Thank you of our speaker.
Podríamos tomar una foto por el día para que tengamos. Si lo que quieras, tomamos una foto cuando